So what I'm going to go through today is not just heat tolerance in cattle. I'm also going to give an introduction to genomic selection and propose it's a technology that we can use both in animals and plants to accelerate genetic gains to meet this big future demand that we have for food. Then I'll go into how we can use genomic selection to improve heat tolerance in cattle and finish up with a bit of a look into the future of animals and potentially plant breeding as well. So before I introduce genomic selection, 50 years of animal and plant breeding in one slide basically. Animal and plant breeding is a very powerful science, and you can see that in the impact that it's had. So I think this is over 30 years of breeding for increased growth rate and improved feed conversion efficiency in chickens. The time to market weight for a chicken to reach, the time taken for a chicken to reach market weight has decreased from 16 weeks to 5 weeks over that time. And here you see a picture of an improved genotype and the original genotype fed on the same diet at 5 weeks of age. So there's a dramatic difference there. And in plant breeding as well, the gains have also been dramatic. And this example is from hybrid corn in the US, where the gains are 77 kilograms per hectare per year, pretty much year in, year out, from selecting improved varieties. So it really, animal and plant breeding really has had a substantial impact. However, we need to go faster to meet this challenge we have of feeding 9.5 billion people by 2050. So how are we going to do that? Luckily, there are some new opportunities, some new tools that we can use. And one of those is genomic technologies. The DNA marker technology has come an enormous way even in the past 5 years. So the cost of getting DNA marker information is now about 1,000 times cheaper than it was 5 years ago. So the SNP chip technology, and you can see these SNP chips up the top of the slide here. These are small glass slides that you can wash the DNA of an animal or plant over, and it will assay the genome at up to 800,000 points along the genome. So this technology is there. It's actually developed for human genetics, but we can use it in animal and plant breeding to hopefully increase the rates of genetic gain. But how are we going to do that? There's a bit of a challenge here as well. You may think that you could use that technology to find the SNP for increased yield in corn, or the SNP for increased yield of milk in dairy cattle. Unfortunately, the traits we're targeting to improve don't have that architecture. There's no single genes with large effects on those traits. And I'll demonstrate that. This is another corn example. And this is a picture from a lovely experiment for selection for increased oil content in corn kernels. That's been running for over 100 years. And on the x-axis there, you have the generation, and on the y-axis, you have the oil content in the corn kernels. And you can see the oil content's just going up generation after generation. And in this paper, these researchers tried to dissect how many genetic mutations were underpinning that response. What they found was there were at least 50 mutations that explained only 50% of the variation between the different lines. So that tells us that each mutation has an effect of just 1% on that trait. And most of the traits we look at, particularly yields, have this type of architecture. For milk yield in dairy cattle, for example, there's actually thousands of mutations, all with a small effect on the trait. So how are we going to improve traits with an architecture like that? We can't just find one SNP or one marker that's going to do the job for us. What we can use is this genomic selection technology, and the technology was developed specifically for traits like this. The way this technology works is you need to start with a large reference population or training set. So you need a large number of individuals that are measured for your trait of interest. Say it's milk yield in dairy cattle, yield in corn, or whatever. So you have a large number of individuals measured. You also genotype those individuals for a large number of markers across the genome. Might be 50,000 markers or 800,000 markers across the genome. Then you put that information into the computer and let it work out the effect of each of those 50,000 markers or 800,000 markers on the trait. And what happens is, although the effect of individual mutations might be small, because you're using all the markers simultaneously, all the markers at once, they can each pick up the small effects of those mutations. So what you end up with is a prediction equation that says I have 50,000 markers. The effect of the first marker on, say, milk yield is an extra gram of milk. The effect of the 50,000th marker is an extra 2 grams of milk, and so on. And you have an effect for each marker which is tracking the effect of each of the genes affecting the trait. Once you have that prediction equation, you're really in business, because what it means is you can go to any individual, say a young calf, get a sample, extract the DNA from that, and genotype it. And then you can predict its genetic merit for that trait. And you can do that very early in the animal's life, even at birth, for example. Once you've done that, you have the genetic prediction, the value of that animal's genetics, and you can make selection decisions based on that. And this is genomic selection. Now, it was really pioneered in dairy cattle, and there was a good reason for that. Before genomic selection came along, the way dairy cattle breeding worked was to work out the value of a bull's genes for milk production. Of course, you can't milk the bull, so you had to wait till that bull had daughters, and then you'd milk those daughters and use that to evaluate the bull's genetics. That takes a very long time. It takes about 8 years before you can evaluate the bull's genetics. With this technology, you can evaluate the bull's genetics at birth. And instead of waiting 8 years, you can start to breed from the bull as young as 18 months. So you can really speed up the number of generations of selection you can do. So that's why most dairy industries around the world, the selection is now done on the DNA marker profiles using genomic selection. And not so long ago, I counted up how many dairy cattle would have been genotyped around the world, and it's now greater than 2 million for the purposes of genomic selection. So it's being used very widely in that industry. It's also accelerating gains in other livestock and crop species as well. So that's an example in dairy, but this genomic selection technology is also really good for traits that we've had trouble selecting for in the past. And traits that we've had trouble selecting for because they're either hard to measure or expensive to measure. And that includes things like meat quality, fertility. I've got an example here in sheep and in cattle, and also feed conversion efficiency, which is a very economically important trait, but very expensive to measure. Now with the genomic selection technology, if you can measure a reference group of animals, you genotype them, you get your DNA marker predictions, you can select any animal out in the industry based on those predictions. So it's great for traits like this. It's also good, I'm hoping to convince you, for another trait that has been really hard to select for, and that trait is heat tolerance. So resistance to heat stress. Heat stress in cattle is a problem. Once cattle go outside their thermoneutral zone, they start to have to expend energy to either cool down if they're heat stressed or warm up if they're cold stressed. And in dairy cattle, that range might surprise you. That thermoneutral zone is between about 0.5 degrees and 20 degrees. Once you go above that, particularly as you go above 26 degrees, the animals are working quite hard to dissipate the heat. And in dairy cattle, the impacts of heat stress, the first thing you see is a reduction in intake, followed by a reduction in milk yield. If the heat stress is really bad, they'll stop lactating. And we've seen this in heat stress events down in Victoria. The lactation just stops. And also there's a very big impact, harder to measure, but there is a very big impact on fertility as well. And in beef cattle, same thing, you get a reduction in feed intake with heat stress, reduced growth rates, fertility is compromised, and also an increased susceptibility to diseases and parasites. As the incidence of heat waves increases, and particularly really extreme events increases, heat stress is a bit of a threat to us, and declines from production due to these increased heat stress events could threaten our ability to provide this very high value protein from both milk and meat for more and more people. So the question is, what can we do about it? What can we do to make our cattle more heat tolerant? One tried and tested approach that works very well, of course, is to use more adapted genotypes. So there are quite clear differences between breeds and between Bos taurus and Bos indicus cattle in their ability to tolerate heat. And some of the mechanisms underpinning that are actually quite well known. There was lovely work done back in the 1950s here at CSIRO, where researchers actually counted up the number of sweat glands in a given area for Bos indicus and Bos taurus cattle. And they clearly showed that Bos indicus cattle had more sweat glands. So, I mean, breeders have been, and northern cattle producers have been exploiting this fact for quite some time now and creating crosses between Bos indicus and Bos taurus cattle. But we could take this even further by selecting within breed as well. It's been demonstrated by quite a number of researchers that there's within breed variation for cattle to tolerate heat stress, and we could potentially exploit this through selection. We could exploit this through genomic selection. The trouble is we need a phenotype to do this. We need to build a reference set so we can work out what our DNA markers are tracking for our heat tolerance trait, so we can start to use genomic selection. And our reference population has to be big because it's quite likely the effect of these genes is small, so we need a lot of animals in that. So how are we going to get a phenotype that is a heat tolerance phenotype on lots and lots of animals? One thing you could do is send out people around the country with thermometers and actually measure rectal temperatures on thousands and thousands of animals. And you could do that, but it's quite expensive. So the phenotype we chose to use in dairy cattle was to take advantage of the fact that Bureau of Meteorology has all these weather stations scattered throughout the country. And what we decided to do was line up herd test day data. So this is yield of individual animals that's recorded a couple of times every month over the lactation of these cows. So we can actually get the yield from individual cows of milk and the corresponding temperature and humidity on the day of milking and a number of days of milking before that as well. So we can see what's the level of heat stress and how much are individual cattle producing. And this map just shows the dairy farms that we had dotted around the country where we had data coming in, and the weather stations that were located close to those dairy farms. So we've got those two pieces of data, temperature and humidity, and the yield of the cows. And if we start to get that over multiple time points, multiple weeks or months over the year, what we can do is come up with a phenotype which is how the cows are responding to increased heat stress. So along the x-axis here, I have an index that puts together temperature and humidity, because it's the combination of those that actually create heat stress. As the humidity goes up, that makes it harder to dissipate heat. So it's the combination that's important. So that comes from the weather station data. The milk production comes from the herd recording that the farmers are already doing. And for each individual cow, we can see how she's responding to that increased heat stress. And what you see is there's a threshold of temperature or humidity beyond which milk yield starts to decline, but some cows decline further than others. So this is our phenotype now. And in this little example, as the heat stress comes on, cow A, her yield is declining less than cow B, so she's more heat tolerant. And this trait turns out to have a genetic component. There is genetic variation there. It's about 10% heritability. So that's our phenotype. Just to show you the number of days of the year that cows are experiencing heat stress in different states, this is after we collected the data. X-axis number of days of the year, and then you've got the states. In Tasmania, not much heat stress really. There's a few heat waves that go through, but not much. In Queensland, you've got up to 50 or 60 days where you've got quite severe heat stress there. The next is the data we had, and the key thing here is the number of cows where we could actually work out this decline in production with increasing heat stress. And there are about 366,000 Holstein cattle, the black and white cattle, milk-producing cattle, where we could work out this phenotype, and about 76,000 Jersey cattle where we could work this out. So quite a large data set. However, we only had genotypes on a fraction of those. And where I've got bulls there, this is actually the average of how their daughters decline. So a bull might have 1,000 daughters, and you can get a trait which is the average of how his daughters decline with heat stress. And we had some individual cows as well. And they were genotyped for 800,000 markers scattered across the genome. So from that genotype data, we lined it up with the heat tolerance phenotype, and we've got this on the cows and also the bulls as the average of their daughters. We can use this to get genomic predictions for heat tolerance. And that's exactly what we did. So we estimated the effect of each of those 800,000 markers on the heat tolerance, and then we come up with a prediction equation. So when you have new animals that come into the system, you can genotype them and predict how heat tolerant they'll actually be. You can do that, but if you're going to convince breeders and farmers to actually use this, you have to have some kind of validation to demonstrate to them that it actually works. So we've done this in two ways. The first way was to split up the data. So we used about 2,300 of the sires, 2,200 of the cows as our reference set. We used that to calculate the effects of each of the 800,000 markers to get the prediction equation. And then we've predicted a validation set of a bit over 400 bulls for the Holsteins. And those bulls all had daughters. on which we had this heat tolerance phenotype. So what we've done is predicted how heat tolerant those bulls should be, what their genetic value for heat tolerance should be, and then we've lined it up with how heat tolerant those bulls' daughters actually were. So we've predicted their heat tolerance and then correlated it with their daughter's actual decline in production. And that's what this number here is. That's the correlation between your prediction and how heat tolerant their daughters actually were. So it's about 0.5. And what does 0.5 mean? For milk production traits like protein, fat kilograms in the milk yield, we're getting up around 0.8 now for our genomic predictions. So quite a bit higher accuracy. But for a new trait that we didn't have information on before, this is quite useful. So that's one way you can demonstrate the accuracy. The other way you can do it is to get two groups of animals, those predicted to be tolerant and those predicted from the markers to be heat susceptible, and then follow them through what actually happens in a heat wave. Does their prediction actually drop off as predicted, and less for the tolerant animals than the susceptible animals? So that's what we did next. And this is our validation study. So in our validation study, we went out across Victoria and selected or genotyped 400 heifers. So we've taken a sample from them and extracted their DNA and run them across this panel and predicted how heat tolerant they should be. Then we actually purchased from the farmers the 24 predicted most heat tolerant heifers out of those 400 and the 24 predicted most heat susceptible from the farmers. The next thing was to take them through a heat wave, and we've done that using climate control chambers down at the DPI Victoria research farm. So they have 6 of these lovely, they're actually methane measuring chambers, but you can also control quite precisely the temperature and humidity in those chambers, so we can use them for this study. So we've taken these animals through a mild simulated heat wave. And we've measured, as they're going through this, it was a 4-day heat wave, we've measured milk production and core body temperature, rectal temperatures. So what happened when we did this? First of all, just to show you what the heat wave looked like, on the x-axis you just have the time of day or time of night. So this is going through the day. And this line here is just the temperature. So it's a bit above, a little bit below 30 degrees, up to 30 degrees, and then a little bit below. So it's not too severe. However, this was done in winter. So the difference between the outside temperature was reasonably large. And this is the humidity that they're experiencing. Right, so what happened? You remember there's these two groups, predicted heat tolerant and predicted susceptible from the markers. And this is what happened to their actual milk yields as they were going through that 4-day event. And what I've got here, this is day 1, day 2, day 3 and day 4. And on the y-axis is how much their milk production declined from the baseline before the heat wave event. On day 1, not terribly much happened because it takes a while for the heat load to accumulate. But by day 2, there was quite a clear difference. The heat-tolerant animals didn't drop their production much at all, whereas the heat-susceptible were dropping by up to 3 litres.
And is that litres per day?
That's litres per day that they're dropping. Yep. So they dropped 3 litres from, they were yielding about 24 litres, so it went down to 21 litres. Whereas the heat-tolerant didn't drop much at all. As the mild heat wave event went on, the heat load does accumulate, and even the heat-tolerant animals start to get more of a drop-off. But the heat-susceptible animals have a larger drop-off there.
So just to kind of day 3, is that an extra 1.5 litres from day 2, or is it?
No, it's all from baseline. So if it was 24, it's whatever this is subtracted off 24.
Day 3, they sort of bounce back.
But what I'm not telling you here is that 3 of the heat susceptible animals had to be removed from the experiment. We had very tight thresholds on things like respiration rate and panting rates. We didn't want the animals to feel distressed, so as soon as they hit those thresholds, they were removed. And it was interesting, 3 heat susceptible animals had to be removed, whereas no heat tolerant animals were removed. So I think that's why they come back closer together there.
And food consumption makes up how much of the drop?
About 60%, I think, 60 to 70%. So it's a lot driven by feed consumption, but not completely. The other really pleasing or interesting result to me was there was a difference in rectal temperatures as well. So the heat-tolerant animals were more able to sustain their core body temperature than the heat-susceptible ones. So here you can see the increase. That's the baseline before the experiment starts, and this is as the heat event rolls on. And you can see the heat-tolerant ones always have a lower rectal temperature. We had another measurement of core temperature, which was intravaginal temperature, and these were little sensors that sent temperature every minute over the Wi-Fi, and you can see again the susceptible are always tracking above the heat-tolerant animals. At the end of the experiment, they start to come together again. But we're seeing these real physiological differences. So what it suggests is we have genomic breeding values that really can be used to distinguish heat-tolerant and heat-susceptible animals. So this genomic breeding value is intended to be rolled out to the dairy industry, to be used particularly for farmers in regions where they have these heatwave events quite reasonably frequently. An interesting question, if you're a farmer, is how do I balance selection for heat tolerance with selection for everything else? It's actually milk yield that pays the bills. Fertility is also really important. Disease resistance is important. How do I balance that thing? So one of the postdocs in the group down in Melbourne that was working on this project actually came up with a little web app that optimised the balance of heat tolerance with everything else. So in this little web app,